Hello friends, in this video we will be talking about cellular events of acute inflammation. In previous video we were done with vascular events. Let's start. Cellular events includes 1. Exudation of leukocytes. 2. Phagocytosis. In exudation of leukocytes include 4 changes. 1. Changes in the flow of blood. 2. Rolling and adhesion. 3. Transmigration. 4. Chemotaxis. Phagocytosis including 3 steps. 1. Recognition and attachment. 2. Engulfment. 3. Killing and degradation. Now, we are detailing more about exudation of leukocytes. In this, leukocytes are escape from lumen of microvasculature to the interstitial tissue. For this here, 4 changes occurs, that is, 1. Change in the flow of blood. In this, there are 2 words that describe this changes, which is margination and pavementing. Now we are going more deeper in this. Normally, in blood vessels, there is 3 things are running. That is plasma, red cells and white cells, that is lymphocyte. Red cells and lymphocytes are flow in center of blood vessels, and plasma is peripheral, so normally plasma is close to the vessel wall. In case there is any injury or due to any causation, we are already discussed in part 2, that is vascular events. In this we understand that increased blood flow due to vasodilation and subsequently, there is slowing or stasis occur. After that we understand exudation of fluid into extracellular space. So now, due to fluid is transfer in extracellular space, blood vessels gets narrow because of loss of plasma. And that phenomenon is known as margination. As a result of this, neutrophils also comes close to the vessel wall. This is known as pavementing. Second change is rolling and adhesion. Due to margination and pavementing, neutrophils slowly roll over the epithelial cells lining the vessel wall. This called as rolling phase. After that, leukocytes are also sticks to the endothelial cells. This is known as adhesion phase. Third is transmigration. In this, neutrophils are move along the endothelial surface. Between the endothelial cells is found where the neutrophils throw out cytoplasmic pseudopods. Subsequently, the neutrophils lodged between endothelial cells and basement membrane crosses the basement membrane by damaging it locally with secreted collagen ases and escape out into the extravascular space. This is known as transmigration. Now move towards 4th change, that is chemotaxis. It is a process in which, after the transmigration of leukocytes are moved towards the direction of chemical molecules. Chemical molecules are of 2 types. 1. Exogenous agent. 2. Endogenous agent. So these chemical molecules are binds to specific transmembrane GPCRs, which is a receptor present on the surface of leukocytes. After that GPCRs send signal for effector molecules on neutrophils. And then these effector molecules act on membrane phospholipids and increase cytosolic calcium. This results in cytoskeletal changes. These changes results in forward movement of the leukocyte towards the chemical molecules. With this we are done with exudation of leukocytes. 2. Phagocytosis. Phagocytosis is cell eating. It is defined as the process of cellular engulfment of a solid particular material, like microbes, foreign particulate materials. And these processes performed by cells called phagocyte. Phagocytes are of 2 types. 1. Polymorphonuclear neutrophils. 2. Circulating monocytes. Phagocytosis involving 3 steps. 1. Recognition and attachment. In this, macrophage having cell surface receptors which recognize microorganisms. After that, microorganisms coated with specific protein, opsonins from serum, and this process is called opsonization. That means preparing for eating. Now that opsonins function are to establish a bond between bacteria and the cell membrane of phagocytic cell. So in this, a bond occurs between bacteria and the cell membrane. Now, second is engulfment. In this, opsonized particle is ready to be engulfed. This is accompanied by formation of cytoplasmic pseudopods around the particle, and this is due to activation of actin filaments beneath cell wall, then enveloping it in phagocytic vacuole. Eventually, plasma membrane enclosing the particle breaks from cell surface, After that, phagosome become internalized in cell and lies free in cytoplasm. Phagosomes fuses with lysosomes and form bigger vacuole, that is called phagolysosome. Third is killing and degradation. In this, microorganisms are killed by antibacterial substances. After that, killed microorganisms are degraded by hydrolytic enzymes. And this is major function of phagocytes, that's why is called scavenger cells. However, this mechanism fails to kill and degrade some bacteria like tubercular bacilli. So with this we are done with cellular event. Summary: in this session we are done with cellular event, under that, exudation of leukocyte and phagocytosis. Thank you.